Hello, everybody, and welcome back to Science and Nature Untapped, a free virtual monthly speaker series brought to you by the River Institute. My name is Stephanie Hildebrand. I will be your host for tonight. So before we begin, we'd like to acknowledge that many of us are living on unceded land and traditional territory of Indigenous people. We are grateful for the opportunity to live where we do, and we thank all the generations of people who have continued their responsibilities to Mother Earth since time immemorial. If you're interested in learning more about our educational programs, our projects, our research, and upcoming events, you can follow us on either Twitter, Instagram, or Facebook at the following handles. And in case you ever wanted to go back and watch a presenter, you can go to riverinstitute.ca/ontap and the links will be there. For upcoming events, instead of having a Science and Nature for May, we'll be doing the River Institute Annual Report 2020, a year of adaptation. This will be a great opportunity to meet our people, to learn about what we did over the last year. This will be May 5th, which is the first Wednesday of May from 7 to 8 p.m. And we look forward to sharing with you all the projects we've been working on. So many of our, well, much of our research, our events, and our programs are sponsored by generous contributors. If ever you are interested in making a donation, you could go to riverinstitute.ca/donate. And of course, you can always just share our social media, and that goes a really long way in terms of support as well. So we thank you for that. So for tonight, we'll be talking about exotic invasion of the Upper St. Lawrence River, Alison Derry's research program, which brings evolutionary biology, community ecology, and ecosystem ecology under the unifying theme of eco-evolutionary interactions. The research is based on aquatic ecosystems across Canada and in Alaska, spanning from the Canadian Rockies to Ontario and Quebec to Newfoundland. So I guess with further ado, it's all yours, Alison.
Thank you so much, Stephanie, and thank you so much for the invitation to present tonight, and a big thank you to the River Institute. I'm just going to share the screen.
Thank you for joining us.
Okay, so the presentation should be up. Can you view it from your side?
Yeah, it looks good from my end.
Okay, excellent, great. Thank you so much, everyone, for joining tonight. Okay, so exotic invasion, that is when species are moved outside their natural distributions, they become established, they spread, they have huge social and economic and ecological impacts. That is the definition of exotic invasion. It is a global threat to aquatic biodiversity and ecosystem function. It's a global problem. It's not going away. There's evidence that at the global level, there is no saturation yet in the spread of invasive species. So it's a big problem, and it interacts with many other anthropogenic impacts, with the resulting effect of loss of biological diversity. And this is a big problem. We're currently recognizing that we're in a period of extinctions and major loss of diversity. And so exotic invasion is a contributing factor, it's an interacting factor, it's a direct result of human activities that's leading to loss of biological diversity. The problem with exotic invasion is that it is difficult, if not impossible, to eradicate exotic species from natural systems. And so that means that it is very important to understand factors that could moderate the impacts of these exotic species once they are irrevocably integrated into food webs, into the native food webs. And so examples of moderating factors that can dampen invasion impacts on native communities are factors such as time since invasion, spatial environmental heterogeneity, intraspecific variation, and adaptive processes in natural populations. And so these are the factors that I'm going to discuss tonight. And as mentioned in the title, the focus of the talk tonight is on the Upper St. Lawrence River and exotic invasion in the Upper St. Lawrence River. And I will be speaking a lot of, the students that have really contributed to this body of work are, in this talk, Louis Estorg, who is a PhD student in my lab, and also Christina Charette, who is a PhD student in my lab and co-advised through the River Institute. And the talk, in particular, we're going to focus in on a particular exotic species called the round goby. This is a fish that was brought in from the Ponto-Caspian area. It was introduced by ballast water from transoceanic ships in the lower Great Lakes. It then spread through the lower Great Lakes and entered the Upper St. Lawrence River and was first detected in Cornwall around 1994. And then at that time, it then rapidly spread through the Upper St. Lawrence River. That is the section that is freshwater in the Upper St. Lawrence River. So you can see in the top panel here, this was the early stages of invasion. I should mention these are Quebec. This is a graph I borrowed from the Quebec Ministry. And so it was introduced, it rapidly spread through these upper reaches of the freshwater section of the St. Lawrence River. And then through the years, the abundance has just kept increasing and increasing and increasing. Year after year, this species is now pervasive through the system. It has had enormous impacts on the food web. And so we're going to focus on the species tonight, but I should mention that its distribution also coincides often with many other exotics from the Ponto-Caspian that are in this system, such as quagga and zebra mussels, among others. And so with the round goby, what happens when it comes into these food webs is that it inserts itself at an intermediary level. So here it is. Here's the round goby at an intermediary level in the food web. And so what it does is it ravages macroinvertebrate prey communities. In the lower Great Lakes, its diet is really strongly associated with exotic zebra and quagga mussels. And a lot of studies have suggested that the exotic invasion of these mussels has facilitated the round goby's establishment. But in the Upper St. Lawrence River especially, its diet is much more diversified. Not only does it feed on these mussels, but on other items as well, such as amphipods, chironomids, gastropods. These are all really important prey for native fishes with whom it directly interacts, or competes rather, and takes away resources from. At the same time, it has become a resource base for pisciferous fish, such as smallmouth bass, yellow perch, and then of course gets into the food web even higher into the aquatic birds through those fishes as well. So it's inserted itself in, it's here to stay, it's pervasive, it's now in the web. And there's little we can do to eradicate it in this very dynamic system. And so this is why it's really important to consider how the natural system itself might mitigate some of these impacts. And so, as I mentioned, one of the factors that can mitigate the ecological impacts of exotic invasion is time since invasion. And so, okay, so just to kind of start off, there have been many studies on the ecological impacts of round gobies, many, but most of them focus on what happens after the first 5 years of detection. There's a few studies that have investigated longer term effects over 10 years. 2 of the studies are in the Upper St. Lawrence River, one focused on the macroinvertebrates, one focused on the fish, but there really aren't a lot. And it is really important to look and track these populations and these changes over time, because the impacts of the invasive species can be maintained or they can change over time. And this is because of evolutionary processes that occur both in the invasive and residence populations, because of changes in the communities themselves in terms of what species are present and their relative abundance, and also if the environment has changed. And so most studies lack a temporal context. Okay, and it's so really important to consider that temporal context. The reason is because we cannot expect any species, let alone an exotic species, just to always continually exponentially increase, okay? Because it reaches a point where the population reaches a carrying capacity in relation to the resources that are available, and those abundances should theoretically stabilize over time in relation to the resources that are available. And so this very first study that I'm going to present is kind of building on those first studies that looked at the initial impacts, building on the subsequent studies that looked 10 years after invasion, to come back even later, 20 plus years after invasion, to look at how sites with different times since invasion, whether the round gobies abundances have stabilized, and how is that in relation to available resources. And so the study I'm going to present, there's a bunch of models, but we disentangled time since invasion from whether, like, the influence of invasion itself, whether presence or absence of invasion. So we'll kind of leave it there. So the part of the river that we focused on is really this upper stretch. It's the upper St. Lawrence River, but it's the upper stretch running from Lake St. Francis to Lake St. Louis. And the round goby is pervasive through the system, as I showed you, but there is sites with a range of times since invasion. Some sites, the gobies have been there a long time, 19 plus years, and at other sites, it's more of a recent invasion event. And even though the gobies very quickly progressed to the system, the times at which they actually colonized these sites vary, and it's really strongly linked to the environmental characteristics of local sites themselves. So you can see that some of the sites that have been invaded the longest are in the St. Francis, the Lake St. Francis here near Cornwall. This is where the species entered the river. The conditions there are very favorable for gobies. So these are some of the longest sites that have been invaded. And then you have even sites that remain uninvaded in the system. And then you have more recently invaded sites, around 12 years and then 13 to 16 years. And so based on these previous studies that recorded the presence of gobies and our own research, we were able to characterize whether sites, the time since sites had been invaded and whether they were uninvaded. And so the first graph that I'm going to show you is on the y-axis, we have round goby abundance. On the x-axis, it's time since invasion. And what you can see here is that there's this exponential increase in the abundance of gobies running up to sites that have been invaded for about 13 to 16 years, okay? They just more and more keep coming every year, just more and more gobies, okay? But what we see here at the 17 to 19 years, these older invaded sites, kind of the first signals that the population sizes might be starting to level off. And so then now going to the resource base, these are macroinvertebrates. And so we have their taxon richness on the y-axis, we have time since invasion on the x. And what you can see is the effects of gobies are enduring across sites of different times since invasion. But at these longer invaded sites, even though there seems to be signals of stabilizing abundances, this is where you really see that the macroinvertebrate communities are just completely stripped right down. These are the sites where the gobies have been present in high abundance for 17 to 19 years. They've ravaged the prey communities. Another graph here, just very, very briefly showing, okay, so these black triangles here, these are the sites that have been invaded the longest. What you can see without kind of going into the details of this graph is that all the families of macroinvertebrates, their abundances, are piled up away from those sites. In other words, again, those sites that have been invaded a very, very long time, they've been stripped of the macroinvertebrate resources. These resources are so important for native fishes, they've been stripped. And where you find these taxa that would normally be present, they are present at the sites that haven't been invaded as long or are uninvaded. In terms of the fish communities, the effects of round goby and the time since their invasion, it's less clear. And that's because gobies can have positive impacts sometimes in terms of providing resource for certain species, and negative impacts for others in terms of competing for resources or, destroying areas of spawning for others. So what we found was conductivity actually really drove more the structure of fish communities in this system rather than the time since invasion. But I'm going to return to this conductivity gradient in later on in the talk. And so just to conclude this first study, with the passage of time, the continued increase in round goby abundances would be expected to decline at invaded sites in relation to available resources. However, just because round goby abundances may stabilize over time in relation to resources, it may not be that the prey resource recovers. It could happen or it might not. And what may happen is that that prey resource is just kept and maintained at a very low level and unable to completely recover. And so it means that we really need to continue monitoring this system in terms of understanding how the abundances of round gobies are changing, because they are changing, and what that effect has on these macroinvertebrate communities that are such an essential resource for other fish in the system. And so when we can kind of look at the ecological effects of other invasive species around the world, what they've shown is that there's actually evidence for enduring ecological effects of exotic invasion on resident communities. And so this other research suggests that even though the round goby abundances may stabilize, their impacts are going to endure and stay with us. And so, as I said, we just need to really continue tracking the system because it's so dynamic, to understand this very delicate balance between invasive predator and very impacted native resource community. So with that, I'm going to continue on to the next moderating factor, which is that can moderate the ecological impacts of exotic invasion, and that is spatial environmental heterogeneity within the system. So what is spatial environmental heterogeneity? So what it is it's variation in the physical and ecological landscape characteristics. And so this, in a broader sense, is very well known for influencing and promoting biodiversity at the landscape scale. And so spatial environmental heterogeneity has a potential role in the preservation of native species diversity and abundances in invaded systems. And it can do this by one, potentially limiting the spread of biological invasions by providing refuges. In other words, refuges that have conditions that are less conducive to the exotics. And so the exotics either can't get in or they're held down at lower abundances within the system. And so most research on the importance of environmental heterogeneity in invaded environments has actually been done in terrestrial ecosystems. And hardly anything has been addressed on this topic in freshwater ecosystems. And the irony is, as this is despite that freshwater ecosystems are among the most invaded ecosystems in the world. And so in the upper St. Lawrence River, there are two potential types of refuges. One is already well known. In the upper St. Lawrence River, there is a broad scale conductivity gradient with different calcium concentrations. And this gradient is a very strong limiting factor of round goby invasion. And that's because the round gobies, as I mentioned, are Ponto-Caspian in origin. They came from the Black Sea, Caspian Sea, and so they evolved in a brackish water environment. And so where they do well in the system is in water masses where there's a higher conductivity, where there's higher calcium. And that water is the water coming from the Great Lakes. It's pouring into the St. Lawrence River, and near the city of Montreal, it meets the Ottawa River. The Ottawa River rolls in off the boreal shield. It is brown water. It's brown water carrying organic matter. It is low conductivity, is low calcium. And that presents a formidable, like a very strong barrier for round gobies and other Ponto-Caspian exotics to get into. And what happens is those two water masses meet. They run side by side, almost as far as Quebec City, at which point the salinity influence from the estuary breaks it up. And so what you have at this kind of regional ecosystem scale is this very broad scale gradient that creates a very broad scale refuge, okay? A refuge where natives can go across both. They can go across the gradient. They've evolved there. They can deal with the low ions, the high ions, and the low calcium, the high calcium. But the round gobies, the zebra mussels, the quaggas, and other Ponto-Caspian exotics, they cannot penetrate it. So those low ion environments are a refuge for the natives. The other type of potential refuge that really was a lot less known about until we did our study was the role of wetlands. And so there was research out there suggesting that gobies, their distribution was not only restricted by things like conductivity, but also by factors such as substrate type, like whether it was muddy or whether there were rocks. And so that created the possibility that wetlands potentially had a really important role for locking out round gobies and protecting native species. And then as a secondary is another thing about wetlands. Wetlands are known also, independent of exotic invasion, to promote freshwater diversity and really contribute disproportionately to regional biodiversity than other water body types. So there was a real potential for wetlands to have a very positive role in the upper St. Lawrence River in terms of protecting native species from the impacts of these exotics. And so we set out to do a study on this. And so our objective was, can different refuge types reduce invasion impacts on native freshwater biodiversity? Again, we focused on the round goby as the sentinel exotic species that represented the distribution of other Ponto-Caspian invaders. And we wanted to look at the effects of environmental heterogeneity created by the broad scale conductivity gradient and also the local wetlands that occurred both in the high conductivity water and the low conductivity water. So once again, here we are focusing on the stretch of the river freshwater section, but not going further than Montreal, between Cornwall and Montreal. And so what you can see here on the south side, Cornwall, this is St. Lawrence River proper. It's where the water is running from the Great Lakes through the system. It's the high conductivity water. It is the invaded waters. To the north here, this is where you have the Ottawa River running into the system. Again, brown water, calcium poor water. It is locking out invasives from coming in. They cannot deal with those conditions. The natives can deal with it all. They've evolved there. It's their system. And where they meet at Montreal, this is where the water masses meet. But as I said, there are mixing zones, but those masses are distinct for much of the river. They run along there. And so as a result, you have these ion poor refuges and you have these invaded ion rich waters, and on both sides you have wetlands. And so we sampled in wetlands and adjacent to wetlands in both of those across that gradient. Okay, so just starting off the broad scale gradient, what you can see here is Okay, conductivity increases on the x-axis and the round goby abundance increases on the y. So what you can see is outside of wetlands, really nice positive correlation between goby abundance and conductivity. That is what I explained to you before. We knew it, we've seen it, but there it is, a nice strong correlation. What's interesting is if you look at the wetland response, which is the yellow line, across that same gradient, just adjacent to off the wetlands, there is no correlation with conductivity. In other words, the wetlands are breaking that relationship. They're tamping down on the goby abundances. They're breaking that positive relationship with conductivity. So on the invaded side, in the high-on waters, those wetlands are there. They are refuges. And then when we compared effect sizes, so Cohen's d, it's like comparing the relative effect of one refuge versus another. What we found was the broad scale conductivity refuge, it is definitely the strongest. It is the strongest. It is at the broad scale, those low ion waters on the north side, they are blocking the gobies from getting in. It's twice as strong. But the wetlands still had a significant effect, and their significant effect came in on the invaded side. In other words, they didn't have much of an effect on the low ion side because it was like a refuge within a refuge. So kind of they're there and performing another role, but not controlling goby abundance on that side. But on the invaded side, the wetlands, again, very important for reducing goby abundances. And then we look at the resources again, back to the macroinvertebrates that were so impacted in the previous study that I showed you. And so what we found running across the conductivity gradient, an increase in diversity across the conductivity gradient, meaning that more different taxa and kind of very evenly spread in abundance. However, when you look at total abundance, it just crashes. And so remember, it's these high conductivity sites are the invaded sites. The low conductivity sites are not invaded. And so this is a signal of invasion impact. Okay, so then we go to the wetlands. And so what we find in the wetlands with these macroinvertebrate communities is that the taxon richness is so much higher in the wetlands compared to out. And same with the abundance. The abundance is higher in the wetlands compared to out. And these relationships, again, are not necessarily driven by the wetlands on that ion poor side that is not invaded. They're driven by the wetlands on the invaded side of the river, the invaded side with the high conductivities. So again, evidence that wetlands really important not only for locking out gobies, but also for maintaining macroinvertebrate abundance and diversity. Less clear for the fish communities what was going on. Again, gobies, depending on the species, can have either a positive or a negative effect. But what we did find was that the number of different species in the high conductivity wetlands was diminished, but the beta diversity was increased. And what beta diversity means is that each wetland had a unique assemblage of fish. So maybe the number of species in the high-on wetlands was lower, but they were all different species. And so those high conductivity wetlands had a very important role for hosting unique fish assemblages compared to out in the open water. And so that kind of brought us to the next question was, okay, so we're seeing positive effects of wetlands on the macroinvertebrate resources. But is it a direct effect of wetland on the communities, or is it an indirect effect through a reduction in round goby abundances? And so we used a technique called structural equation modeling to be able to distinguish between these direct and indirect linear effects, kind of using this multivariable approach. And what we found overwhelmingly was it was the direct effect of wetlands that was pumping up the aquatic biodiversity in the macroinvertebrates and in the fish beta diversity, as I mentioned. And so just to kind of pull that together in terms of these first two papers that I've presented, number one, round goby impacts on the macroinvertebrate resource communities accumulate with the passage of time where they have invaded at these high conductivity sites. So that was the first paper that I showed you. But now the second paper gives some hope. It tells us that the low conductivity waters, and which are uninvaded, and the wetlands in the invaded high conductivity waters, these are refuges from an exotic invasion. They're refuges because they either exclude or they reduce goby abundance. And what that means at the ecosystem scale is an overall dampened effect on exotic invasion. It has dampened the ability of these exotics to really get a hold on the system more so than they already have. The wetlands, especially in the invaded high conductivity waters, these have the role of enhancing macroinvertebrate diversity abundance, hosting unique fish assemblages, and so overall the effect is a pumping up or an enhancement of biodiversity at the ecosystem scale. So working together, wetlands suppress exotics and they pump up the native biodiversity. And so what that says is that it's so important to preserve wetlands in the upper St. Lawrence River, especially in the invaded high ion waters in the St. Lawrence proper channel. It is essential for helping to mitigate invasion impacts. Okay, so now we are going to move to the third mitigating or moderating factor of exotic invasion, and that is intraspecific variation and adaptive processes in native populations. Okay, so intraspecific trait variation, that is variation within species. It enables adaptive responses in response to the environment that might include things like human impact, anthropogenic impact, such as exotic invasion. It can act on it. It's just both of these are sources of selection that act on the variation within species. It can provoke adaptive responses in populations. And what that means, and I'm going to explain various adaptive processes that can occur, one of which is evolution, but it means it's not only adaptive response and evolution for that species, but what it means is those adaptive responses can scale up. They can scale up to affect ecological processes in communities and ecosystems. And this is a new field called eco-evolutionary dynamics, where we try and understand the interplay between evolution in populations and ecology in communities and ecosystems and how they feed back on each other. Okay, so in the context of exotic invasion, when we think about adaptive responses in native species to an exotic invader, what you can have is an exotic predator come in, and the native prey evolves a defense in response to that predator. And so there are studies out there, for example, European green crab on the Atlantic coast, that's the exotic predator. Native prey snail evolves defense to try and deal with that new predation pressure. That's one example. Another example possibility is that you have an environmental gradient, such as in the St. Lawrence River, where the exotic predator can only occupy part of that gradient, but the native prey can slide and occupy the whole gradient and potentially evolve adaptations that facilitate its ability to coexist with the predator. Okay, so that is the situation that we're going to talk about today in this last moderating factor. And so what type of adaptive responses can occur? Okay, so going across an environmental gradient, you might have an adaptive response called plasticity. Okay, what does plasticity mean? Basically, it is the adjustment, the phenotypic adjustment of an individual. In other words, an adjustment of metabolism, an adjustment of appearance. It doesn't involve any genetic change. It means that if an individual is taken from one environment, it's placed in another, it can change its metabolism, it can change its appearance to deal and optimize its survival in the new environment. And so if you have that occurring in a species across an environment, you just have a lot of flexible individuals who adjust their phenotype according to what environment they're in, where they find themselves on the gradient. The other type of adaptive response that you can have is called local adaptation. This is a genetic response. It usually entails less dispersal across the gradient. And what it means is that individuals in the population in divergent environments on different sides of the gradient, they optimize their phenotypes to that particular environment. They're optimized, but they're not flexible. In other words, for example, perhaps this is a high ion environment, and so they're completely locally adapted to high calcium conditions, whereas on this side, they're adapted to low calcium conditions, as opposed to being a flexible type that can move across both and deal with both conditions. And so that if you move an individual from this side to this side, it's maladapted. It's genetic. It can't really adjust unless there's some kind of new adaptation that occurs. And finally, a third adaptive response that can occur is a maternal effect. And so maternal effects are a form of, basically it's a form of plasticity, but it's mediated by the mother. And so what it means is a mother in her environment transmits information to her offspring. And so the offspring has a similar phenotype as the mother, because the mother has given the offspring some information about her environment that's adaptive for the offspring. And so, great examples are resources. A mother that provisions, who's flush with resources and provisions her offspring with a lot of resources, the offspring have a lot of resources. But that effect might change in the next generation if the offspring during their adult life have impoverished resources. So it's a bit of a lag effect. There's a generation lag by one time, by one generation, in which the plasticity kicks in again. So it's kind of a little bit intermediate between plasticity and local adaptation. It is not a genetic effect. Okay, so we're going to consider these possibilities for native species that occur across this ingredient and how it might help them coexist with exotic species. And now really focusing in on Lac Saint Louis, which is that part of the river where those two water masses come together. So rolling off the Great Lakes, coming in high on water, and on the north side, low ion water coming in from the Ottawa River, there's mixing. And so we looked at divergent environments on other side of the same system where there's, again, high potential for dispersal across these gradients, but very different environments. The native species can occur across both, the exotic species only on one side. Okay, we are now going to move from gobies to amphipods. What are amphipods? Amphipods are a type of macroinvertebrate. They are a major component of fish diet, including the goby. And so when I showed those impacts on macroinvertebrate communities, that we're talking a lot about these guys. And the story thickens because in the macroinvertebrate community, we not only have native species, but we also have exotic macroinvertebrates, and those include exotic amphipods. And lo and behold, those exotic amphipods, they came from the same region as the gobies. They can only also exist on that iron-rich side coming from the Ponto-Caspian Black Sea area. And they compete with the native amphipods who can occur across the gradient, who evolved in the system, who are adapted to it. But we are going to focus on these native amphipods in terms of how they're dealing with this system and using their adaptive abilities to be able to coexist with predators such as gobies and exotic amphipods, which actually attack these guys when they're molting. Anyway, so here we are. So we're going to look at those different adaptive strategies that I mentioned to you in the iron-rich versus the iron-poor habitats for these macroinvertebrates. And I'm just not going into detail on these experiments, but these experiments were very complicated, and there were two types. There were kind of two different parts of the experiment. The one experiment focused on field-caught individuals. We collected them in the field, we brought them to the lab. They were exposed to either iron-rich or iron-poor water. These responses, if there were differences, reflected plasticity. Plasticity, why? Because there was no genetic change. It was the same individuals brought in from the field, they put in the lab in contrasting water, and if they changed their phenotype, it was an individual changing their phenotype without genetic change. So we interpreted that as plasticity. That's all you need to know about that one. The second experiment involved actually breeding them, making those field-caught amphipods have babies and bring them into a second generation. Why? Because we wanted to know, okay, what happens if we get rid of the plasticity, and is there kind of a signal of genetic effects on either side of the river coming in, okay? So that's all you really need to know, is then we took those babies and exposed them, well, not babies, we reared them till they were adults to control for stage, but that offspring, that next generation, to see if there were differences, and then we inferred those if there were differences as genetic or maternal effects. That's all you need to know on this. And so just to kick it off, the first trait that we looked at was post-molt calcification. So this is really important. Amphipods, to grow, they have a carapace. To grow and to reproduce, they have to get rid of their carapace because it's like a little armor. They get rid of it, and then they do their growth, and then they draw calcium from the environment to rebuild their armor. The thing is while that armor is off, they're super vulnerable to predation from those exotic amphipods, okay? So what we found was these native amphipods, the black, okay, so this is percent calcium body content. The black points are from the ion-rich side. The open points are from the ion-poor populations. Okay, so we brought ion-rich guys in, put them on ion-poor water. Guess what? Whoa, less calcium in their bodies. They slowed down. They just tracked the water. So that was what we call co-gradient plasticity. But what happened with the guys coming from the ion-poor water? We put them in ion-rich water. They went crazy. They put on more calcium than you would expect at a much higher rate. This is called counter-gradient variation. It's a compensation. It's like they ramped up their metabolism way more than the individuals living in the ion-rich water to pack on that, the richness of calcium in their water. So this was very interesting evidence of plasticity, very adaptive. If you imagined an ion-poor copepod making its way over to the ion-rich side, which is invaded, very quickly rebuilding its armor following casting off the old one. So that's very interesting and very adaptive. The second trait we looked at was development time to reproduction. Now, these results are for the offspring. So these results are findings where we had effects that were maintained into the next generation. Didn't matter if you changed the environment, they were just there. In other words, it was genetic. And what we found was the ion-poor amphipods coming on that boreal shield side, that brown water, they took forever to develop, twice as long as the amphipods in the ion-rich invaded water. So just really long development times. And so you might imagine that if an amphipod from the ion-poor water side made it across to the invaded side that's ion-rich, this is not an advantage because the individual just develops so slowly, it would make it extremely vulnerable to predation. However, this went along with higher survival and higher fecundity in the high ion-poor amphipods. So again, doesn't matter what water they're in, that's just the way they are. Higher survival babies, more babies, even though they have longer development times. So longer development times, not good, but that investment in development time comes out in babies with higher survival and more of them. So this is a good thing. So it's like those bundles of traits go together, and what one might seem as a disadvantage is actually an investment in the other. So what that brings us to when we think back, okay, we've got a gradient, we've got ion-poor refuges on one side, we've got ion-rich invaded sites on the other, we've got the exotics on the rich side, the natives going across. What does that mean? Okay, so it means that in terms of post-molt calcification, natives moving across the gradient, they can adjust their phenotype, they can do it quickly, and they can do it better than their conspecifics living on the rich side. That is a big advantage. However, they develop slowly, not good. However, with that slow development comes more babies and higher larval survival. And so these were the results that we found. So what it comes to is different adaptive strategies for different traits. And where it becomes important is when we think about, okay, what does that mean? We've got natives sliding across the gradient using different adaptive strategies. They meet the exotics that they have to deal with. What does it mean? What does it mean in terms of net influence on demographic subsidies? In other words, are these low ion refuges dumping subsidies, demographic subsidies on the invaded side, helping to maintain the species persistence at a whole in that system? Noting, of course, that in the lower Great Lakes where we don't have these refuges, these natives have been kicked out and displaced by the exotics. And then, so what that means is perhaps because of it, we have the enhancement of coexistence between the exotic species and the native species. Okay. So there we go. So we've talked about moderating factors on ecological impacts of invasion in terms of time since invasion. Populations can't keep growing exponentially forever. In terms of spatial environmental heterogeneity, in terms of providing refuges from low ions and wetlands, and also adaptive processes in native populations, helping them to coexist. Where do we go next? So one thing I really alluded to was really the importance of trying to understand the changing abundances of native species in relation to, of exotic species in relation to native species in the system. And one tool that's coming to the front for environmental monitoring is environmental DNA. So I'm going to talk a little about that because it's hit the press. What is it? What are the caveats? How could it be applied in this system? So what environmental DNA is that it is DNA that is shed by the body of organisms into the aquatic environment. It means that one only needs to go and take a water sample from the environment, extract the DNA from that water from a filter, amplify it, and then sequence it. There's two different approaches. One is called meta-barcoding that identifies multiple species in a sample, but it is very complex to interpret. It can be. There's a lot of bioinformatics involved. And the other approach is a single species PCR where you focus on a single species and you actually quantify its abundance. So there's those two different approaches that are out there. And just kind of bringing you into another invaded system where we started to do some research on this new tool, kind of thinking about how it might be applicable to other systems, is the Rocky Mountains. So in the Rocky Mountains, brook trout are an invasive species. They're native here. They are valued here. They're valued by recreational fisheries, okay? But in the Rockies, they were planted into historically fishless lakes. They were planted for the purposes to stimulate tourism in the Rocky Mountains. They are extremely destructive. They displace native trout. They ravage food webs. And so we were given the go-ahead to go in and harvest entire lake ecosystems. Harvest them not only for their removal, but also to test some theory on size-selective fishing for research. But it also opened the door to test tools about environmental DNA because it meant that we knew exactly how many fish were in a lake. And that is very rare to have that information. And so we did some research in which we had very confident estimates of the density of brook trout, the biomass of brook trout, and then we were able to scale the distribution of masses of individual fish, their sizes, in relation to the amount of environmental DNA in the water. And what did we find? Really nice correlations between abundance and environmental DNA concentration in the water, especially if you take into consideration differences in body size and the distribution of body size in fish populations. And so this could really be applied potentially to the St. Lawrence River system to understand how round goby abundances are changing, for example. It's so hard to make those estimates on these natural systems. And so this is potentially a very powerful tool for that. However, there are caveats. That previous study that I presented to you was done in closed lakes. In the St. Lawrence River is a highly dynamic moving river system, it means that you have a complexity of other processes occurring, such as transport, deposition, as well as degradation that occurs in the lakes as well. However, although eDNA can travel in these moving water masses, most eDNA is deposited at a small scale, which is promising, but it needs to be flow corrected if we use this as a tool for comparing differences in abundance, for example, of different fish species. Presently, eDNA has been applied in the St. Lawrence River for presence-absence of invasives. It has not yet been applied to estimate abundance. It has not yet been applied to do metabarcoding to estimate changes in community diversity, but the potential is there. Okay, so pulling in towards the end, we talked about the importance of understanding factors that could moderate the ecological impacts of exotic invasion once these exotics get irrevocably integrated into food webs. And some of these moderating or mitigating factors are understanding the influence of time and stabilizing abundances of exotics in relation to resources. The role of spatial environmental heterogeneity in terms of providing refuges and having positive diversity impacts at the ecosystem level scale. And also the potential for native species to have adaptive responses and adaptive strategies that help them to coexist with exotic species. In terms of moving forward, as I said, tracking those population abundances of invasive species in native communities that might be facilitated with emerging tools such as environmental DNA, but also integrating other frameworks such as meta-community approaches. What is a meta-community? It is populations and communities that are connected by dispersal. And I alluded to that with the movement across the gradient of that native and whether it's providing demographic subsidies from refuges to invaded sites. Also integrating evolution, eco-evolutionary dynamics, that interplay between the adaptive response in natives and how they're interacting with those invasive communities. And finally, food web science, trying to understand how exotic invasion, such as by-round gobies, has changed nutrient provisioning and also contaminant bioaccumulation in the food web. And I won't get into that because that is something that Christina Charette is studying, and perhaps in 2 years, she'll be here in this seminar series to present that research to you. But at the end of the day, what's really important to take home is the importance of conserving uninvaded refuges. So whether that is like these low ion refuges on the north side of the river in Lac Saint Louis, but also really, really important, the conservation of wetlands in the invaded waters, because this is how local communities and people can really play a part in they themselves helping to mitigate these invasion impacts in a very dynamic system such as the Upper St. Lawrence River. And so with that, I would like to acknowledge the many funding agencies that the research in my lab is supported by. I would like to thank, of course, the students in my lab that are the engine that does the research across the diversity of these systems that we study, including the St. Lawrence River. And I would especially like to thank you for joining tonight. So with that, I will hand it back to Stephanie.
Thank you so much, Allison, for taking the time to share your know-how, your experience, and your research with us on a topic that I think many of us are familiar with to a certain extent. So thank you very much. I'm just going to say goodbye to everybody who's watching from your TV, because I know we have a bit of a time limit. So thank you again for tuning in. Next month, we'll be having the River Institute annual report. So we're very excited to share that with you. And so have a good night and thank you. So for everybody who's watching online, it's a question period, and I'm just going to open up and see. So we have a couple questions that have come in, so I'm going to publish them and I will read them to you, Allison. So the first one is from Patrick from the South Shore of Montreal. He says, you're talking a lot about the limitation of the abundance of the invasive species by the abundance of its resources. How's the importance of predation on these species as a growth limitation factor? Is it an important factor or is it negligible?
Okay, so yeah, so that's a very good question. And there isn't research out there to address that question directly. So I'll just have to kind of use my knowledge. What we do know is that the round goby has become a very, very important resource base for fish such as yellow perch and smallmouth bass. And so actually Christina is studying how that resource base is providing nutrition to them, and if there's variation across the system with these gradients in terms of how the round goby is providing nutrition, but also whether it's changing pathways of contaminants, because with the round goby, it's changed the system from kind of a pelagic system to a benthic system because it feeds on mussels, because it feeds on a lot of these macroinvertebrates. Is it making it a more efficient transfer of methyl mercury up the system to these higher fish? So that's like a consumption concern to fishers. Whether those fish are regulating the goby, we don't know. They are regulating it to some extent, clearly, but given that we found that very that like kind of exponential increase of gobies, up to about 16, 17 years that they've been at a site, and then it drops off kind of at the 19 years, those pestiferous fish would be at all those sites. And so to me, that drop in goby abundance is probably related to the devastation of the resource, and because those resources have been so heavily stripped. So I would think probably they're more regulated by a bottom-up resource base than by the pestiferous fish that are feeding on them, as well as native fishes. I hope that helps with the question. But again, there's no study that has actually specifically validated that question, but that's just my inference from the research I showed you tonight.
Great, thank you. The next question is from Brian Hickey. He says, you focused a lot on benthic invertebrate community changes, but is there evidence that other native fish species with ecological niches that are similar to round goby are being impacted?
Yeah, okay, yes, there are. And yes, so I mentioned that the goby impact on the fish communities is more scattered. And so there are fishes that are negatively impacted. And so fishes such as darters, so benthic fish that feed on similar resources or live in very similar habitats as gobies, such as darters, those are negatively affected. And we did see negative correlations with those. But then with other fish, it was really less clear, especially when we were comparing wetlands and off of wetlands, because there are more gobies outside of the wetlands. Were those gobies outside the wetlands drawing fish from the wetlands to go out and feed on them? So yes, we found effects, but it was not as severe at the community level as on the macroinvertebrates. And if you look at a species-specific level, some effects are positive, some effects are negative, such as for the darters.
Great, thank you. Thank you for your question, Brian. And we got a comment from somebody named Erica Crispo. She says, thank you, Allison, great talk.
Oh, thank you, Erica.
And so next we have Dave. He's asking, what is your next area of research?
Oh, what is the next area of research? So that's a great question. And yeah, that's a really great question. So in the St. Lawrence River, I think it would be very interesting to look at these meta-community dynamics that are kind of facilitated by the physical continuity of the system, but yet you have these divergent environments. And so we've started to do some genomics work, for example, on some of the native species to try and understand if there are signatures of selection on their genome, how much migration is occurring across the gradients. So that's definitely an area of research. And then, as you know, we are as I mentioned, we are doing some of the food web work with Christina and the contaminants and the nutrient provisioning. But one emergent contaminant that's kind of hitting the river are microplastics. There's a lot of interest to look at microplastics in the St. Lawrence River. I would do that in collaboration with contaminant people, but microplastics may be really facilitating bioaccumulation of other contaminants absorbed to the plastics, and gobies may be a big part of that story because they've become this important part of the resource base and because they kind of feed on the bottom. So I think that's another emerging area for the St. Lawrence. And then, yeah, so I'll leave it in the St. Lawrence. I have other programs happening in other parts of Canada that are opening up, but I'm definitely happy to talk about them, but perhaps they're for another talk.
Great, thank you. Thank you, Dave, for your question. Next up, Philip Ling from Maitland Tower asks, were any constructed wetlands examined for effectiveness as refuge?
Yeah, that's a great question. And so far as I know, they weren't. And that would be a really great follow-up study to distinguish between, they were not just, maybe they were by Louis, I'd have to ask the student, but he definitely did not distinguish them or recognize them as that. And so I think a really great follow-up study would be in comparing the wetlands, the natural wetlands that were kind of always there versus ones that people have actively tried to restore and their effectiveness. So that would be a really, really great follow-up study, and it would be a wonderful way also of integrating the community as well in understanding kind of the direct effects, like the direct positive impacts of their efforts. But we didn't in our study, in the one that I showed tonight.
Great, and we have another follow-up question from Philip Ling. He's saying, we are in Maitland, about 100 kilometers upstream of Cornwall, a mature round goby habitat. Has been a huge increase in cormorant populations to the point of calling for culling. Maybe this is part of the reason for the decline in the gobies there, and if culled, the gobies would come back?
Possibly, yeah. I think that's possibly, yeah. I think that is a really, really interesting observation. I've very aware of the cormorant issues, and I've watched them myself in the Great Lakes, boom and bust. And I think that probably they're really fast increases. I think that that's a great inference that it's linked to the goby. But I think, yeah, I think if there is a cormorant call, if they are doing a top-down regulation of the gobies will come back. They will come back because they're not gone. They're not eradicated from the system. And they can always recolonize from the Great Lakes because it's this open system. And so it'll just tamp them down. And you might even have compensatory growth, in which case you get even more gobies than you had before, possibly.
Thank you. I know the cormorants were a hot topic in Ontario, so it's nice to hear a little bit about that.
Yeah, yeah. Yeah.
So Patrick, so Patrick, he was saying you answered his question perfectly, and to remind you of the question that he had asked, it was, you're talking about the limitation of the abundance of the invasive species by the abundance of its resources? How's the importance of predation on these species?
Yeah. So that's great. I was thinking about the fish, but I wasn't thinking of the cormorants, but that's like a really, really great segue. But I mean, thinking about that too, upstream of Cornwall, those are some of the longest invaded sites. Those sites are like among the first that were hit by gobies. They had the highest abundances. They've been there the longest. And so their resources are also probably like the gobies are just keeping those resources tamped right down. And so now with the cormorants coming down and reducing goby abundances, has it been long enough for macroinvertebrate communities to recover? I think unlikely. And so then now, so in terms of gobies coming back, they'll come back. But again, I think they're going to be limited by that eroded resource base in terms of the level that they come back.
Thank you. We have a question from Matt Windle and a comment. So he says, I really enjoyed your presentation, Allison. I was wondering if you had observed any differences in body condition of gobies in wetlands versus non-wetlands as a further sign of dampening or additional to abundance.
Yeah, so that's not something that we measured, at least by the studies that we've published. It's something more that Christina is measuring. She is measuring body condition, and she's very interested in how goby condition and other native fish condition changes across these gradients. And she's using traditional techniques such as weight and length, but also more emerging techniques such as fatty acid profiles. And so I would say that's research that's forthcoming through Christina's research, and that she could probably integrate wetlands as a part of it. She was really focused on these broader gradients, such as nutrients that are pumping up algae in the system versus the dissolved organic carbon, the brown waters, that tends to dampen productivity. But I think the wetlands would be interesting for her to look at too.
Great, thank you. I have a question from Brian. He's asking, is road salt, which influences conductivity, going to have an impact on the invasion of round goby, or is it specifically calcium that is important?
Yeah, so that's a really great question. So there's two things. So first of all, the lab studies not done by my lab, but by another lab that have correlated this conductivity gradient to reduced goby abundances have linked it with the calcium specifically. And so road salt, when it's added, is usually sodium chloride. So the gobies, it wouldn't solve the calcium problem, it would increase the conductivity. And so I think it would actually, if there is calcium going in with the road salt as well, I think that it would make the goby problem and the Pontio-Caspian invasion problem worse. Yes, I do believe that. Other questions arrive because like these fish evolved in brackish waters, right? So if you bring kind of more of a brackish situation, they're going to do well. Whether they can, but now they've kind of colonized this freshwater system. They can maybe spread with road salt, but they actually might be limited at the ocean end because now that they've adapted to freshwater, they may not be able to recolonize, further than Quebec City. So that's like another question. It's like a salinity question, not road salt, kind of taking it at the other end. So short answer, yes, I think it'll make it worse. And actually road salt is something that our lab is also studying as well. Right now, we haven't crossed it with the exotic invasion problem, but that would be a very nice step forward.
Thank you, Allison. I have a question from Noah Picut. He says, hi, Allison. I just had a question about wetlands and how they help reduce the invasion of the round goby. I was wondering if future invasions, since the St. Lawrence experiences some of the largest numbers of invasions of the world, will jeopardize this safe haven, damaging the biodiversity of the wetland and therefore facilitating the round goby to move into wetlands.
Yeah, so that was a question that I had when I started this project, first of all, whether the round goby is adapting to live in ion-poor water, and also whether it's adapting to kind of move into these kind of browner waters. And we did not find any evidence of that there was kind of any change in distribution. And the main features of the wetlands that seem to be kind of dampening down the round gobies is the muddy substrate. It's that muddy substrate because the goby does really well on cobble and rock. And so that's why, like, if people are hardening their shores for recreational purposes to put in rocks and probably make it recreationally nicer to access the water, it's actually really, really enhancing the invasion problem. Whereas if you have a muddy shore, that is the thing. It's that thing that is stopping the gobies from spreading. And then as a result, they're not necessarily going up, for example, up muddy tributaries and stuff like that. So it's that feature of the environment. Whether they're going to evolve to be able to do that in the future, you know, especially with propagular pressure that's adding, you know, all this evolvability into their system because of like coming from different sources and this big genetic diversity pool piling in there, giving them huge evolutionary potential. Whether they will in the future, we just don't know. We don't know whether they'll be able to penetrate or not. But right now, they're not.
Great, thank you.
Question? Yeah.
Let's see. The next question is from Dzie Filippo from Point Claire. He says, why are gobies keeping out of wetlands, even in invaded areas? And I suppose you maybe touched on that, but I'll let you.
Yeah, that's exactly it. It's that bottom substrate. It's the bottom substrate. So in other words, like, yeah, so it's the bottom substrate, which is held in place. It's that muddy substrate that is held in place by the aquatic plants, and the aquatic plants are what make up the wetland. And then because the plants are there, it becomes a deposition zone, and it's like a cycle, right? Like, so so that's what's locking it out, is the muddy substrate. In terms of the biodiversity, the native biodiversity that's enhanced, that is probably like a ecosystem complexity question, where it's like there's structural complexity because of the plants, where fish can hide, and it tends to be a more nutrient-rich area for certain species to feed in. So that would be why the native biodiversity is pumped up, and the properties of the bottom substrate is what's kind of keeping the gobies away, that mud.
Yeah, I've definitely, I work with Matt Windle on fins during the summertime, and we definitely see much less gobies when the substrate is that soft, kind of muddy, yeah. We have, so Brian is asking, in the PCA analysis, all the benthic, oh wait, no, that was, I think he pressed enter for finishing. So let's see. In the PCA, you showed all the benthos were clumped together except mosquitoes. Any idea what that means?
Yeah, so what it meant was, like, in these sites that were, so what it means when you look at those graphs, it doesn't mean that the taxa aren't there. So it doesn't mean that. It means that you can have taxa there, but they're in higher abundances. So they're more strongly associated with sites with shorter invasion times and weaker invasion impacts is what it means. And so the only group that was kind of correlated in where they had higher abundances than in these lower impacted sites were mosquito larvae. Which says something. So those are the only ones that were in higher abundances at these heavily invaded sites than the less invaded. Those other species might have been there, but just in low abundances. That's what it means.
Great, thank you, Allison. And I think maybe we have one more question, and we'll see if we have a flow that comes in, but I think it might be coming to an end. So Noah Picat is asking, the amphipods that had the plastic response to sequester the extra calcium when placed in the ion-rich water, was that extra protection enough to protect it from goby predating on them?
Yeah, so that's a good question. So not extra predation, not extra protection from predation from the goby, because the goby is going to gulp down an amphipod whether it has its armor on or not. So, but what it's protection from are the invasive amphipods, because what happens is those natives are molting, those exotic amphipods come and they eat them. They're like, your armor's off, you're my dinner. And so it's called intra-guild predation, where you have predation at a same trophic level. And so that's what those amphipods are vulnerable to when their carapace is off, and if they take longer to get it back on, that's what they're vulnerable to. But the ion poor guys, as we showed, they hit the rich water and they snap that armor back on twice as fast than they would have in the environment they evolved in. So.
Thank you, Noah Picut, for your questions. I think that's it. There's been like the flow has stopped, and I haven't had any new questions in the last couple minutes. So I guess thank you so much again for taking the time to present. Sorry, I keep losing my camera here. Sorry about that. and I imagine we'll be seeing you again soon. And I guess I should also say goodbye to everybody who's watching us on Teams. So thank you for joining us again. And we look forward to seeing you next month, which will be the annual report for the River Institute. So you'll be seeing a few of us from the RI who will be explaining what we did for 2020. So thank you, Allison.
Thank you so much. It's been a real pleasure being with all of you tonight.
All right. Good night, everybody.
Night.